I would like to introduce, he's not a doctor, he's a scientist, Dan Turner. He's a senior director of applications at Oxford Nanopore Technologies. His responsibilities include developing laboratory and bioinformatic methods to allow users of the technology to perform a wide range of analyses. The applications group also works with external collaborators to develop and demonstrate new uses of the technology. Actually, he's going to show us how to miniaturize sequence machines.
Thank you very much. I realized when I wrote that biography that I didn't really say anything about what Oxford Nanopore is or what we do, so I'm going to start off by, by correcting that. So Oxford Nanopore Technologies is a biotech company, and we make sequencing devices and sequencing-related devices. And people use these for a whole range of different things. So, for instance, people have used our device to look at the microbial community on the International Space Station quite recently, for instance. They've used it to look at plant species up a mountain in Wales, to look at lung cancer expression profiles, and so on. And a few labs, including my own, have used the device to do some proof of concept work looking at PGS, and that's what I'm going to be talking about today. So I'm going to start by explaining how the technology works and what makes it different from other sequencing technologies that you might be familiar with. I do want to say that I'm not a salesperson. I'm going to try and avoid making this talk salesy. I'm a scientist, so we're going to stick to the facts and results and so on. So first thing is, what is a nanopore? And as the name suggests, it's just a very, very small hole. And over the last maybe 3 decades or so, the academic community has shown that nanopores can be used as very, very versatile sensing devices, tools. And they've shown that it's possible to use nanopores to quantify things such as TNT or recreational drugs or proteins. And about 20 years ago, a patent was released that talked about using nanopores to sequence DNA and RNA. And that's what I'm going to be concentrating on today, although not the RNA side, although we can do that. I'll be talking about DNA. So if any of you have ever done any electrophysiology work as part of your studies, you'll be familiar with the device on the left, which is called an axo patch. And this is, it's a patch clamp amplifier. So it's a very, very sensitive, state-of-the-art machine that can measure very, very small changes in current. Now, what you can do with this is you can connect it up to a small chamber. And in that chamber, you can put a lipid bilayer. And in the lipid bilayer, you can put a single protein nanopore. On either side of that bilayer, you can put a sodium chloride solution so that it can conduct electricity, and you can apply a potential across the bilayer. When you do that, the current can flow through the pore, and it can only flow through the pore because the lipid bilayer is a good insulator, and that's represented by the figure in the middle. So the membrane is the black bit, the salty buffer is the light blue bit. And when you do this, this is when the maximum current can flow through the pore, as shown by the trace on the right. We call it the open pore current for obvious reasons. If you add a small molecule to one side of the membrane, and for the sake of argument, let's say it's a negatively charged molecule so that it wants to go through the pore, then as it's passing through the narrowest part of the pore, which we call the barrel, the light grey part, it's going to choke off that current to some degree. So that results in a slight dip in the current. If we put in a larger molecule, it will choke off the current to a greater degree, as shown by a greater dip in the current. So this is the basic principle of how the sequencing technology works. When it comes to looking at nucleic acids, it's slightly more complicated because there are many ways that a strand of DNA can interact with the amino acids in the pore than just on the basis of size. So there's no guarantee that, let's say, a small nucleic acid will cause a small block and a big one will cause a big block. But the point is that every time a particular base goes through the pore, it will cause the same block, and we can learn what that looks like, and we can work out how to interpret it. The other difference is that when we have DNA going through a pore, it goes through intact. The strand has many, many nucleotides joined together. And the barrel of the pore can accommodate about 5 different nucleotides at the same time. Now, if you remember that there are 4 different bases, that means that in each of those positions you can have 4 different things. So there are about 1,000 different states that we have to be able to distinguish between, which can exist in the barrel of that pore. What this means is that when we are taking the electrical measurements, we get a signal that looks like the thing, well, this is a real example of a strand of RNA passing through the pore. It's not human readable by any means. You just see a squiggle of lots and lots of different current levels, maybe 1,000 different values in there. And we have to work out what all of those are. And there are some things that you can kind of measure. Sorry, that's not what I wanted. There are some things that you can kind of make out. So, for instance, if you look to the left, there's a flat section, which is the poly A tail of the RNA going through. And to the left of that, there's an adapter sequence, which if you've seen these before, you can recognize because we have to attach an adapter to the strands to get it to go through at all. But generally, it just takes a lot of very complicated mathematics to turn that signal into letters. And I'm not going to spend my time explaining that. Now, it's all very well doing sequencing on a single channel pore, like you would get by connecting up an AXA patch, but you can't really scale it. And if you want to generate the kind of genome-wide data that you would need to do PGS, then a single pore just doesn't cut it. But you can't really parallelize an AXA patch because it's about the size of a VCR machine. So over the last 10 or 12 years, the company we've a company made of physicists, chemists, electrical engineers, biochemists, just a lot of different disciplines coming together to miniaturize this technology and to parallelize it. And a few years ago, we released our first product, which is called the minion, or minion, which is shown on the left. It's a handheld device. It's maybe about the size of a mobile phone. And this has 512 nanopores on it, on the consumable, on the flow cell, which means that it can sequence 512 strands at the same time. But it's important to point out that then when one strand has gone through, this pore is then available to capture the next one. So if you leave it running, you'll capture thousands, hundreds of thousands, or millions of strands. So you can get a lot of reads from this thing. It's not just 512. So there are a few really key components to the technology. The obvious one is the one that comes up in the name again and again, is the nanopore. This is just an example of one called hemolysin. It's not exactly the one we use, but it illustrates the point. But the nanopores, we can engineer these because they're proteins. We do a lot of site-directed mutagenesis. And we're looking for nanopores that have got a few very key qualities. We need a nanopore that's stable so that we can ship these things around the world and keep them in our storeroom. We also need it to be able to accommodate a single strand of DNA, not a duplex, into the barrel. The membrane also has some key features. I mean, the main feature that the membrane has to have is that it's got to be a very good electrical insulator so that the current can only flow through the pore and nowhere else. And the third component shown here is called a motor. So if we would just put DNA into the top of the pore and let it go through on its own, it will go through at a rate of something like a million bases every second. And our electronics aren't fast enough to measure that. So what we have to do is slow it down. So we use a protein that is capable of grabbing onto the DNA strand and effectively lowering it through the pore one base at a time. As it does so, it pauses slightly, and that gives us a split second in which we can take lots and lots of current measurements. We're sampling at several thousand hertz, so we can take many current measurements and work out an average. The motor protein also has to be able to unwind a duplex ahead of it, which it does. And it's also tunable, so we can make it go faster or slower by adding more or a somewhat lower concentration of the cofactors that it needs. And the final piece of the puzzle is what we do with our sample. Because we can't just take some DNA that we've extracted from a cell or whatever and put it into the device and have it start sequencing. We have to do something. Well, we can do that, but the number of strands we'd see would be very, very low. So what we do is we attach some DNA adapters to the end of our fragments, and they've got some, again, as with everything else, got some really key properties that without which it wouldn't really work very well. So at one end, this is very much like trying to thread a piece of cotton through the eye of a needle. We put a piece of, it's not working, but we put a piece of DNA on that's got very low secondary structure, so that instead of it being bunched up in a hairpin or whatever, it's available for threading into the pore, and we call that the leader strand. Every adapter has got the motor protein preloaded onto it, and that's just an efficient way of making sure that everything that gets the adapter has the motor protein on. But we don't want that to be working its way along the strand until we're ready, so we block its movement with another piece of DNA. It's a very complicated adapter structure. And the final thing is that the membrane that the pore sits in is hydrophobic, whereas DNA is hydrophilic. And so the two don't want to interact, but that makes the sequencing very inefficient. So what we have to do is put another piece of DNA with a hydrophobic molecule onto it. so that when that template finds the membrane, it attaches to it and it diffuses in 2 directions rather than 3. This has the effect of making the sensitivity about 10,000 times better than it would have been otherwise. So if you put all those things together, you make a DNA sequencer that's got a few unique properties. And they are, I mean, the one that we're probably the best known for is being able to create very long reads of sequence, so tens of thousands of nucleotides consecutively, up to hundreds of thousands. When that might not seem like it's directly relevant to PGS, because you're generally dealing with quite short fragments, but it can be in some circumstances, and I'll explain why in a little bit. The other thing is, because the sequencer doesn't use optics, there are no lasers, there are no cameras, it's really small. So as you can see in the picture, I mean, that's a real photograph of someone holding one, so it's a portable device. Now, you might not be interested in taking your sequencer to wherever you're doing the testing, but another way of looking at it is that it takes up very little room in your laboratory. It's also a low-cost device, so I'm not going to kind of bang on about that too much. But you can ask me at the end afterwards if you like. And the final thing, which is not immediately obvious, but is when the strands are going through the nanopore, they go through intact. What this means is we're constantly measuring the electrical signal, and we're doing the base calling in real time. So as the strand's going through, we're working out what the sequence of that strand is. And this means that we can make some kind of feedback loops for the analysis. So we can be monitoring how much data we're generating and which bits of the genome we're covering. And then we can keep running until we see the right amount of data that we know we need to answer the question that we set out to answer. So we call this run until. And that means that when we get the amount of data we set out to achieve, we can stop the run and we're not wasting any time. Another feature which is probably less relevant here is something called read until, which means that we can be sequencing the strand as it's going through, and we can be checking whether it's the thing that we wanted it to be. So, for instance, if we're sequencing contamination, or we're trying to do something targeted and this is off target, we can eject it from the pore and move on to the next strand because it will capture the next one, but possibly less relevant here. Again, I didn't really say very much about what I do, but here's just a slide to kind of illustrate it. I like to think of applications as having 3 pillars. One of these is sample extraction and library preparation. So my team's responsible for working out the best ways of getting DNA or RNA from whatever sample it might be, whether it's leaf, stool, blood, whatever, and then turning that into a format that's compatible with sequencing on the minion, attaching those adapters. We have lots of different options available because your priority might be that you want to get to the answer very quickly because you're trying to identify an infectious disease, or it might be that you want very, very long fragments because you're trying to assemble a genome, and we would treat those differently. The second strand is bioinformatics, because as you've seen, the data that we generate and the base calls are different from those produced by other technologies, which means that the tools that you would use to analyze our data are not necessarily the same ones that you would choose to use to analyze someone else's data. So it means that we have to go through a lot of the available software and find out which ones do the best job on our data. And the third pillar is that we put these together along with any unique features of the technology, and we try and think about what projects could we possibly do that would illustrate in the kind of highest profile way to the widest range of people the kinds of things that they might be able to do on the technology if they were to buy it. And this could include more or less any area of genetics or genomics, as kind of illustrated by the bottom of the slide. And one of the projects that we turned our attention to was PGS. I'm in a bit of trouble. There we go. So I'm not here to try and explain why you should or shouldn't do PGS. That's really not something I want to get into. But what I'm going to say, this is the workflow that you're probably all quite familiar with. After a few days of growth, we can take a couple of cells, we can extract the DNA from that, we can sequence it. The trick comes in the analysis, really. So what we have to do is we have to build up a picture of what euploid looks like. So to do that, we have to sequence many euploid samples, and we align the sequences back to the reference genome. And from that, we can build up a picture of what the range of variability within euploid looks like. So then when we get an unknown sample that turns out to be euploid, we can see that the read numbers that hit across the genome fall within the boundaries that we've defined. And so this is shown by the figure on the left. So we can see that we've got 2 copies of all the chromosomes apart from X and Y. We've got one of those, so it's a male and it's euploid. But similarly, if we get a sample that comes along that's got an abnormal number of chromosomes, then we can see that we have an excess number or too few reads hitting a particular chromosome, and we could identify that and call it as aneuploid. So here we've got an example. I mean, this is where we have an extra copy of number 13 by the look of things. And we can, from experience, tell whether it's extra 1 or extra 2 copies or whatever. Right, so I suppose the thing that I should be addressing is not why to do PGS, but why should you use nanopores to do PGS? There's a few reasons that I can come up with, and maybe some of them are more convincing than others to you. That's for you to judge. But this is a typical workflow that I got from Google. And the thing that strikes me is that the PGS test of this part takes about 14 days. Now, because we've got a real-time sequencer, we can really cut that down to the point where we can take the cells and we can return the results within the same day. Now, whether or not that's of any use to people doing PGS or not, I don't know, but there's never any harm in getting the results back quicker. So, one, we can get the results back faster. We have a low, I don't want to try and make any comparisons, but we have a low cost per sample, and like I say, I'll go into slightly more detail about that later, but not very much. I think one thing that, for me, strikes me as a good reason to use a small technology is that you can have these in your laboratory wherever you are, so you don't need necessarily to send your samples off to a testing lab and you then wait for them to get on and sequence the samples and return the data. It means you own the whole process from start to finish. But I think possibly just the most convincing reason for me is that if you aren't already doing PGS, it's cheaper to get going with our device because it's about $1,000 to buy one of the sequences in the first place. So we were convinced that this was an interesting proof of concept experiment to start getting into. So we got in touch with Simon to see if he was interested in working with us on this. And Simon said that the PGS stuff was all very well, but it would be interesting to look at this gene called ANXA5. And the reason for this, I mean, this is a placental anticoagulant, and there are 2 haplotypes. So you've got your wild type, and you've got an M2 haplotype where you have 4 SNPs in fairly close proximity. But this affects coagulation, and it is associated with an increased risk of miscarriage. So my understanding of the situation is that if a couple come along to the fertility clinic with a history of recurrent miscarriages, then both of the want-to-be parents will be tested for their ANXA5 status. And if either of them is found to carry the M2 haplotype, then upon conception the woman would be given daily injections of heparin because this reduces the risk of miscarrying. Now, as geneticists, we know that there's a 50% chance of that embryo having inherited the M2 haplotype from a carrier. And so it means that in at least in 50% of those times, the woman's getting injections unnecessarily. So it would make sense to test the embryo rather than the parents. So as a first kind of thing, this is what we did. We thought we would just go for a very straightforward approach of PCRing across the region, sequencing it, and showing that we got results that matched the capillary sequencing of the same region. And you would hope so for a sequencing company that makes a DNA sequencer. That's what we managed to do. So the results at the bottom show some real results that have been validated by capillary. Blue represents wild type, green represents the M2 haplotype. So most of the cases where the sample was heterozygous, most of the cases where the sample had M2 were heterozygous. There's one homozygous case. You can see that there's a bit of an amplification or some sort of bias towards wild type here, but we sequence to enough depth that that isn't a problem, that we can spot it if it's there. So up until this point, we've been doing the PGS analysis and the ANXA5 haplotyping as 2 separate assays. So we'd get the DNA, we'd divide it into 2, we'd do the ANXA5 test on one, we'd do the PGS screening on the other, and then we'd combine the results and then work out how the sample looked. But we thought it might be quite neat if we could combine that into a single tube assay. Or not exactly single tube, but a single prep assay. So that's what we set out to do. And it was the reason that these things are done separately is because for PGS you want to have low coverage across the whole genome, whereas for a single gene you would want to have high coverage across a very limited area. And these seemed like 2 very incompatible ways that you would need to prepare the DNA. But the answer for this came, it was kind of obvious in hindsight, but normally when you're trying to look at a specific region, you would pick that out by doing PCR. And you'd normally amplify for so many cycles that at the end of the PCR, that's all you can really see if you analyse what's in the tube. So we reasoned that if we did the PCR for fewer cycles, then we could amplify just enough that we saw that target region, but the rest of the region would still be there because you put genomic DNA into the tube at the start of a PCR, right? So that's still there. So if we then put our adapters onto everything and sequence it, and if we get everything optimized right, we can get all of that data out in a single run, which is a very efficient use of the sequencing reaction. So this is what we did, and this worked fine. And it's worth pointing out here that the single gene test that we did for ANXA5 could be adapted for any single or even multiplex gene test if you wanted to. So let's say you had a family history of Huntington's disease or spinocerebellar ataxia or something along those lines. It would be possible quite easily to tweak that PCR part of the test and so you could then do aneuploidy screening as well as the triplet expansion. And this is why the long read capability might be useful, because some of these repeats could be several thousand bases in length, and you might not get that by using shorter reads. So I couldn't come up with a fancy way to display the ANXA5 haplotypes. I've just put them in a fairly conventional table. But these are some real results where we did the combined assay on the samples. So at the top we have the aneuploidy results. These were confirmed by array CGH, so we get complete concordance. So on the left we have a female with trisomy 16. Turned out that this patient, or this sample, was homozygous for the wild type ANXA5 allele. On the right, we have a sample that has a single copy of chromosomes 15 and 16 and was heterozygous for the ANXA5. And we did maybe 20 or 30 samples, I can't quite remember, but we got complete concordance in every case for that. So that was nice. So one question that I've not dealt with yet is how much data do we need to generate per sample to be able to do this test? And I'll stress now that we did not take mosaicism into account. I don't know if we had any mosaic samples. I'm assuming we didn't. So all of this would be the easiest use case. But rather than asking how much data we need, we really should be asking how little data can we get away with and still get a robust answer for the aneuploidy screening. So our sequencer can generate something like, in a good run, 15,000 million bases of sequence information, so 15 gigabases. Human genome is about 3,000 million bases, so 3 gigabases. So if we were to just sequence a human genome on our sequencer, we would see every base roughly 5 times, so we get 5-fold coverage. But we don't need anything like that level of coverage to do aneuploidy screening. We calculate that we need something like 50,000 reads to be able to call additional or 1, 2 few copies of chromosomes very, very robustly. So about 50,000 reads. And because of the way these were prepared, the reads are about 500 bases long for this. So we're talking about 25 megabases of information per sample. If you do the maths, that tells you that you would, in theory, be able to fit something like 600 samples together on a single run, if you were to label each one uniquely. But I'm guessing that for a clinical assay, no one's going to want to do that, really, because there's a slight chance of getting the samples mixed up. So what are we doing to address that? Well, okay, you might remember that I said earlier we can be monitoring how much data we're producing from a sample as the run's going on. So if instead of sequencing 600 samples at the same time, if we sequence one, we could sequence it for 600 times less time, which means that we can do the sequencing part of an assay for this to give us a whole genome screen at that level of coverage in about 5 minutes of sequencing time. So that's all very well, but if it means that you've still got to run one sample on one flow cell, then it's going to cost you a whole lot more. So, we're aware of this, and we're aware that we need to reduce the cost of each sequencing run. And so one way we've got of doing this is to do what is depicted on the right, which I'll need to explain. So the main cost of our sequencing is in the consumable, in the flow cell. And the reason for that is that because it has an application-specific integrated circuit on the underneath of that, and that controls all of the nanopore. It controls the application of voltage. It measures the current and the USB transfer of data and all sorts. We can put the integrated circuit onto an adapter, which means that the flow cell that you would use doesn't need this expensive component on it, and it can then be very, very cheap for a flow cell. We're talking kind of tens of dollars rather than $1,000. So which means that a single sample per flow cell becomes kind of much more realistic. Okay, so what next? I think this is kind of overlapping a little bit with something that we heard touched upon before. So what my team is here to do is to try and find as many different interesting use cases or uses for the technology as we possibly can. One thing that we feel that we can do using this very analysis is to look at higher resolution than whole chromosome duplication or deletion effectively. And as we heard in an earlier talk, there's a class of diseases that I've got a particular interest in because I spent several years as a postdoc looking into these, but microdeletions and duplications. And these are maybe 1 to 5 megabases of consecutive DNA. It's too small for FISH analysis. And although the likelihood of producing an embryo with one of these deletions is very low, the consequences can be absolutely devastating. So they can result in disorders such as Williams syndrome, Angelman syndrome, and there's just many of them. But the problem is that you can't test parents for them because these are spontaneous. They happen during meiosis. So one of the next things we're going to be doing is looking at higher resolution at some patient samples from people with described disorders, and to make sure that we can get that proof of concept to show. But you can see that it would be something that could easily be incorporated into a PGS test with maybe a slight increase in the amount of data. Okay, that brings me to the end of everything that I wanted to talk about. It remains for me to thank a few people, particularly Simon, without whose help we could never have got started with this project, but also to everyone at Oxford Technologies, especially Cecil Yule, who's here in the audience, Owen Harrington, who isn't in the audience, but both of these people really drove the project. And I know I said that I wasn't going to do anything salesy, but I'm a company man after all. And the final word of this is that if you've been interested in what I've had to say and you want to find out a little bit more about the technology, then please check out our website. It's nanoporetech.com, where we have a lot more information about the technology. Thanks very much. I think we've got time for a few questions.